I'll be switching gears now and trying to sort of show one possible way of bridging the gap from the human condition to model systems where we can study the disease using human cells instead of animal models and see if we can get some new insights. This work I'm going to talk about was done in NCBS in Bangalore. I've since moved on to found a new institute in Kolkata, which is focusing entirely on translational neuroscience. The example I'll talk about today is that of Fragile X syndrome. The leading monogenic cause of autism and intellectual disabilities is caused by a mutation in a single gene called the FMR1 gene, results in an abnormally high production of proteins, and it's more prevalent in boys than girls. And the range of symptoms listed here, and my past work and the current work I'm going to talk about has focused largely on anxiety and seizure symptoms. Now, Fragile X syndrome and other such disorders have been studied extensively using animal models, mostly preclinical rodent models, and these have focused entirely on the neuronal, synaptic, and molecular basis of the pathophysiology of the disease, and has given us great new insights into identifying new targets that can be drugged. And this is an exciting area of science. However, sadly, these don't translate, these findings eventually when these targets are taken to trial in humans, they don't translate very well, and more often than not, they result in a very prolonged, expensive, and eventually mixed results at best. As a result of which now major pharma companies are pulling out of this area altogether because for them it's not good business. Yet the problem remains. And what we have found over the last several decades, that despite very exciting new findings in neuroscience, such as Fragile X syndrome, these don't translate into therapeutic interventions of the kind that really works in the real world. And Fragile X syndrome, unfortunately, has become a poster child of this. So what we have learned from this process is that humans are bad models for rodents. And we need to now work on model systems that are of human origin, and maybe that will mitigate some of these challenges that we are facing from translating from animal models to humans. So I'm going to give you an example of that, work done by 3 very gifted people in my lab at NCBS who have now moved on. Shreya is now a postdoc in Edinburgh. Rakipal and Bharat Reddy have moved with me to Kolkata. They did the experimental work, and I must acknowledge Rishikesh Narayan's input in the thinking of this project, and at pivotal points he helped me with new ideas of experiments. So I'd like to acknowledge him as well. Now, this work involves fairly standard protocols of generating iPSCs from human fibroblasts that are eventually programmed to become cortical neurons in this case. These are patient-derived cells, and to complement that, we of course now it's standard to have CRISPR gene-edited control lines as well from ESCs. Together they constitute the kind of set of cells one studies in this model. And in this particular study, we had 2 control patient lines, 3 Fragile X lines, and then 1 gene-edited line. I will interchangeably refer to the Fragile X lines as FXS, the disease lines, and the controls all together. They all show the same phenotypes, and so I'm not going to break them up into these subclasses. And then, along with these cortical neurons derived from these patients, we also generated astrocytes, human astrocytes, and they were eventually mixed together to have a co-culture wherein you have both the cortical neurons and the astrocytes together in a system that has some remarkable properties that I'll talk about. And just to give you a glimpse of what these look like, the gene-edited lines, the control line, the 2 patient lines, and these are the co-cultures shown here, and they lack the FMRP, the Fragile X mental retardation protein. Now, all the data I'll show you is often of electrophysiology, because I do not understand anything that has anything to do with molecular biology. So I'll stick to my lane. And what you'll see today is these cortical neurons co-culture with cortical astrocytes, and the recordings I'll show you are entirely from the cortical neurons, although you'll see that the astrocytes turn out to be the stars of this story. So if you now record from these neurons in these co-cultures, you have basically 4 combinations. You have either Fragile X neuron or control neuron, Fragile X astrocyte, or control astrocytes. In a 2x2 grid, you have 4 combinations, and we'll talk about all 4 combinations. And now for some astrology. So the first thing we observed is that if you have a co-culture of healthy control cortical neurons with control astrocytes. The control neurons fire action potentials in the following pattern. Now, action potentials are spikes of voltage spikes that are the currency of communication that Sudipto briefly mentioned about in terms of synaptic transmission. It's the action potential that arrives at the synaptic terminal and that releases the transmitters that Sudipto was talking about. So this is the currency of communication between neurons. And what we see in this control neurons with control astrocyte condition is that the cells fire bursts of prolonged action potentials, but then between the bursts, there are long intervals, time intervals. This is a healthy firing pattern. Now, here is a cell that's also genetically a control healthy cortical neuron, but it's firing very differently and an abnormal pattern compared to this. It's firing bursts of action potentials which are brief, and there are too many of them. Okay? Here you had a few prolonged bursts. Here you have too many bursts which are all individually very short. But this is a control neuron. Why is it firing like this? It's firing because it's in the company of a Fragile X astrocyte. So a control neuron, just because it's in the neighborhood filled with Fragile X astrocytes, it now starts firing completely differently. If you now take a Fragile X neuron with a Fragile X astrocyte, you also see this aberrant firing pattern where there are too many bursts and individual bursts are too short. And finally, the interesting part of it is you now have a Fragile X neuron in the company of a healthy astrocyte. And although this is a Fragile X neuron, it's firing like a normal neuron just because it's in the company of a healthy astrocyte. So this is quite remarkable that despite being genetically a Fragile X neuron, it's firing normally, which means just adding the healthy astrocyte changes its behavior back to normal. So you can do a correction by just using healthy astrocytes. So what it means is that if you have control neurons with control astrocytes or control Fragile X neurons with Fragile X astrocytes, you see these two aberrant firing patterns. However, if you have a Fragile X neuron with control astrocytes, they fire just like these. And conversely, if you have the opposite combination, these fire like this. So it's the genotype of the astrocyte that determines the electrophysiological firing of the cortical neuron, right? It's not the genotype of the neuron that determines the electrophysiological phenotype of the neuron. So we quantify this, of course, the two parameters, the duration of the burst and the interburst interval. How sparse are these bursts? Those are the two parameters, fairly simple. And we then plot them. You can see that the control neuron with control astrocyte has fewer bursts, and that changes when you add the Fragile X astrocyte, and conversely, the duration can also be modulated. Duration and numbers can both be modulated. And each case, this bar's property is determined by the color of the astrocyte. You can do the converse. So if you go this, there's no difference between these two, despite the fact that they're both Fragile X neurons, but it can convert based on whether they're in the company of the astrocyte. And you can do the same the other way around. Fewer bursts, longer duration can happen in this case as well. So essentially, there's something coming out of the astrocytes that is able to regulate how the human cortical neurons will fire. Now, to probe the underlying electrophysiological mechanisms, and because these are electrophysiological phenotypes, it gives us a huge handle on this, because the underlying ionic basis of these firing patterns are well known from the literature. So we focused entirely on what kind of potential ionic channels could underlie such firing patterns. And based on the previous literature, we honed in on something called the persistent sodium current that has been shown in mouse models of Fragile X to regulate rhythmic burst firing patterns. And we thought we should go after this and see what happens. To isolate this particular persistent sodium current, one has to hold the cells at a negative potential, bring it down, and then ramp it up, as shown here, 20 millivolts per time, and then go back all the way up here and bring it back. So this ramp is a way of revealing the persistent sodium current. So what you do is you first inject this depolarizing ramp in the neuron, and record. Then you use TTX to block out the regular sodium current in the same neurons. You hold on to that neuron and do the ramp again now after TTX and see what happens. And then you subtract this trace from that trace, and what is left is the persistent sodium current trace. So you'll see examples of this. So this is now the control Fragile X combination, and you can see that the control neurons show this tail current, and with Fragile X that is completely missing, the flat red line. So this is the difference. This is the persistent sodium current, okay? In the Fragile X condition, that's completely missing, goes missing. In the control condition, you have this tail current. And you can average these recordings, and you can see there's a huge difference in the current density. In the Fragile X condition, it's completely abolished in the Fragile X condition. And one can do this with the cross combination of control neuron, Fragile X astrocyte, and vice versa. You can see the same phenotype. So this current clearly is getting impaired in the Fragile X condition in all of the 4 combinations that we have looked at. So far what we have is normal firing, aberrant firing, and the persistent sodium current is normal here, and it's impaired in the Fragile X condition. So the question, of course, is that we have isolated 2 different phenotypes. Are they linked to each other? So one way one can do this is to see if we can somehow reverse this impairment of the persistent sodium current by enhancing it pharmacologically, and would that then rescue the firing pattern phenotype that we are seeing? So it's a pharmacological experiment. So there's a drug called veratridine which does exactly that. It opens up the persistent sodium current channel, and so we use that because it's been used in the past in mice. So here now is an experiment where you just add veratridine to the bath, and you see that there's an impaired persistent sodium current, and that's rescued back to higher levels as you see in normal cells. And you can see that as well in this other cross combination, the same thing. So veratridine does rescue the impaired persistent sodium current in the human neurons, and this is the first time that anybody showed this. So now the question, of course, is, okay, veratridine is expected to do this, and it is doing it in human neurons, that's fine, but does it rescue the actual phenotype? So here again, we do it within cell, the same cell comparison. You first establish the abnormal burst firing in the Fragile X condition, then you wash in veratridine for 10 minutes, and it wash it out, and sure enough, you begin to see the normal burst firing pattern again within the same cell. And these are then quantified here, and I won't bore you with the details. And you can do it in the opposite combination as well, control neuron, Fragile X astrocyte, and you can get the same rescue back. So this then tells you that if you use a drug that can reverse the impaired persistent sodium channel current, you do rescue the phenotype, okay? So we have a target now. So the summary so far is, by enhancing the persistent sodium current pharmacologically, we can restore the firing patterns. Then came an interesting bifurcation point where I thought of doing an experiment that was rather unlikely. We wondered if you really need the astrocytes in the co-culture in contact with the cortical neurons. What if we just take them out of the circuit altogether? What happens then? So here now what we're doing is we're growing the astrocytes separately, we're growing the neurons separately, and at some point, instead of adding the astrocytes to make the co-culture, we're just adding the conditioned medium from the astrocytes into this and go on with the same experiments. So now the astrocytes are physically replaced by whatever they're secreting as a soup, and put that into the neurons and see if that's enough. This is what I call the Russum experiment. And so the astrocytic Russum is good enough for these neurons to grow on their own. That was confirmed. And then came the big one. You have a control neuron immersed in Fragile X astrocytic condition medium. So the astrocyte's missing, just the soup is there, and it can make this healthy neuron fire like a Fragile X neuron, as shown here. You can do the converse, which is the therapeutic intervention angle. You can take a Fragile X neuron, immerse it in healthy controlled astrocytic medium, and it starts firing normally. And you can see the same transitions in burst duration and burst number that I had shown you earlier. So this is doing the exact job that you'd want you to do in terms of correction. So what we have now is these two conditions are the boring ones. They do what you expect. But the interesting one is that with Fragile X control neurons, Fragile X astrocytic medium, they convert. And most importantly, Fragile X neurons, just by washing them onto, you wash in the control astrocytic medium, and it fixes it. So then the question comes, what is there in the astrocytic racem that's really fixing this? Now, this is, of course, a big question, because this racem, like the real racem, has a lot of ingredients, and going through them systematically is quite complicated. Some of them are necessary for the growth of the neurons. So again, we sort of, rather than, it's a needle in a haystack problem, and we again resorted to an electrophysiological sort of angle. Our argument was fairly simple. Whatever this candidate is being secreted in the racem from the astrocyte should be of astrocytic origin. It should be able to recapitulate whatever veratridine did. It should do the same thing in terms of correction. And it should have some known properties for modulating neuronal firing patterns. So it turns out, and this is where Rishi's help came in, there's a paper published with mouse, a mouse model system, where similar properties in a different context were shown to have, where they identified S100 beta, which is of astrocytic origin, to do some of the things that we are hoping it would do. The actual effect in the mouse model was opposite to what we were hoping, but we still tried it to see if it works. So again now, this is the question now. Will it act on the persistent sodium current? And if so, will it correct the abnormal firing pattern? So the same kind of experiments. Now, instead of washing in veratridine, you wash in S100 beta onto a co-culture of Fragile X neurons with Fragile X astrocyte. And indeed, the impaired persistent sodium current shown here in the flat red line is rescued. You get the normal current back. This is the average of those findings. The cross combination, you get the same thing as well. So S100 beta is doing the job in terms of reversing the impairment of the persistent sodium current. So that's the first step. Then the question, of course, is would it also rescue the firing pattern? Because we have found this correlation with veratridine earlier. Would S100 beta do the same thing? So get baseline recordings, wash in S100 beta, and then see what happens in the same neuron. And so here is an abnormal firing pattern. Wash in S100 beta, you start getting more prolonged bursts. And if you do the other way around with control neuron Fragile X astrocyte, you get the same. You rescue. This S100 beta alone is able to restore the normal burst firing pattern in the Fragile X cortical neuron condition. So S100 beta is indeed doing the job, and it is of astrocytic origin. So in summary then, what I've shown you is that in the human Fragile X condition, for the first time, we're able to show that there's a cell non-autonomous effect in Fragile X cortical neurons, in the sense that it's the genotype of the astrocyte that determines how the cortical neuron will fire, whether it will fire abnormally or normally. Further, we showed that you can replace the astrocytes with their conditioned medium, and you can do the same rescue. And then we went on to identify one particular molecule secreted from the astrocytes, S100 beta, does the same job. Okay? So one of the things that I like to point out is that we had done similar experiments earlier in an earlier study where we had used human cortical neurons co-cultured with mouse astrocytes, controlled mouse astrocytes. We got the same firing patterns, but although those astrocytes, the mouse astrocytes, were controlled, healthy astrocytes, they were unable to rescue the abnormal firing in the Fragile X cortical neurons, which means it's only the human astrocytes that's able to do this rescue, not the mouse astrocytes. Further underscoring the importance of using model systems of human origin. Okay? So sort of going back to the bigger picture then, much of the Fragile X work for the first 20 years, our work included, focused entirely on synaptic plasticity, aberrations in synaptic plasticity as the key underlying cause for Fragile X syndrome. And these were in rodent models. More recently, groups of people switched to intrinsic plasticity and network activity in mouse systems to show they do play a role as well. What we have introduced now is that we've gone to a co-culture network system that includes both cortical neurons and cortical astrocytes, and shown that not everything is lying within the cortical neuron. There are ways of doing this in terms of cell non-autonomous effect and giving us these astrocytes as a new potential target for therapeutic intervention. So the game changes now. We can look for new ways of reversing defects in the neurons by acting on the astrocytes. So I'd like to thank the folks who've done the work, our collaborators at Edinburgh, funding from the Department of Biotechnology, and Simons Foundation. Thank you very much.
So the question is the therapeutic intervention. At what stage do you think this will be effective? I mean, you've given us enough evidence to show it works,
Right.
but considering it's already a neurodevelopmental defect, so
That's right. Yeah. So that brings up a more fundamental question, which of course you are much more aware of this literature. In the mouse models, the huge deficit, and we're guilty of this as well, is that we look at adult phenotypes in a neurodevelopmental disorder, and very little has been done in terms of the progression.
Sure. Yeah.
Right. So I've taken this back to the rat model now, trying to map out the progression. But in this case, what we see is that these neurons are not really mature neurons, like truth be told. They're very young neurons. But within that, we find that you need at least 8 weeks in vitro for these neurons to mature enough to show these firing patterns. And if you go too far, the astrocytes are so overwhelming that the whole thing lifts off. So you have a very narrow window. So I think the only way to answer your question is to really map it from early on, except we have a limitation that the electrophysiological phenotype is only showing up that 8 to 10 week window, which limits us. So I think we'll have to go back to the drawing board and come up with other phenotypes, more molecular in nature, that can circumvent this problem. But it's a key question that you asked, yes. And we don't have a clean answer to it yet. Yes.
Hi, Shona. Wonderful work. I was wondering, the S100 beta is a calcium sensing protein.
Right.
And, do you suspect that it's directly activating the sodium channels, or is there some other pathway that it follows?
Thank you for asking that question. It is something that Professor Balram pointed out very early, 5 years ago. And so we have done experiments where we have actually played with the concentration of calcium in the medium, and we can switch the firing. So it's clearly S100 beta chelating calcium to some level, optimal. And that must then go through the BK channel or something to modulate. How persistent sodium current channels are linked to that is a big mystery. And it's an intensely researched area. It's not very clear. There are different hypotheses. But for us, we need to first figure out S100 beta's action there, and why didn't the mouse one work? And there seems to be differences in a few in the sequence of S100 beta in that calcium binding domain in mouse versus humans. And that's something we're starting to look at this summer. Yeah.
So is the concentration of S100 beta different between Fragile X and control? Is
yes,
that what is
I had
drawing
that
the
slide. I took it out in the interest of time. It is lower in the Fragile X astrocytic condition medium. We also did a couple of other studies. We also tried to puff on S100 beta onto the cell, because you just don't know what concentration is reaching it when it's in the medium. But those are difficult experiments because they wash away very quickly. So even with the puffing, we could rescue the phenotype for a little bit, but there might be better ways of doing it in terms of actually genetically manipulating S100 beta in the astrocytes, which might be a more stable way of doing it. Yeah. But there is a lowering of the concentration in the medium. Yes. Go ahead.
It's an interesting talk, but I have a small doubt. So in an overall picture, when healthy astrocytes media is taken and it is injected to the neurons, it regains its firing ability. So is it possible that if we filter out the CSF of many of the proteins which are immunogenic and then inject it to a person who is affected, so will those small molecules help regain the metabolic dysregulation which caused the disease?
Yeah. So I think S100 beta was picked entirely because of my bias towards electrophysiology. One has to go systematically and screen for what other potential molecules could be involved, and I'm sure there are. IL-6 is one that we're looking at. And so that has to be done more systematically. We'll just take one last question from Professor.
Yeah, nice talk. There are two questions. One, you are probably aware that astrocytes, they do exocytosis as well.
Yes.
I mean, this has been
Yeah.
shown by capacitance measurements. So do you think this is secreted by the astrocytes? I mean, actively?
I think exosomes would be the way to. So we have to look at the. Yeah, we have to analyze that. We haven't done that yet.
The second question, I mean, you are aware that, when you do a neuron astrocyte culture, the astrocytes proliferate like hell and beyond a certain
yes.
point. So do you have a cutoff time or?
10 weeks, 8 weeks, enough to keep it stable.
The S100B proteins interaction with calcium
Yes.
is unlikely to be involved here, because generally S100 proteins will act in concert with other molecules,
Yeah.
such as annexin beta or even TLR4. So did you probe the role of other receptors?
No, we don't. We don't, and I'm the first person to acknowledge that there are other cofactors we have to look at. The problem is with electrophysiologists, we're stuck with certain restrictions on that, and there's certain experiments we cannot do yet, but those are on the list.